Okay, before I forget. And so, do you have any feedback for me regarding sapling? Have you been working on sapling? Anybody wants to say something?
I had a question.
Yeah.
So I had a question about a couple of the questions on the homework about the vitamin C and the LD. Oh gosh, I don't remember what it was. I couldn't really find much about that in the book or in the slides. I just wasn't sure where we're supposed to get that information.
Well, usually if it's not in a book or on the slides, it's something for you to think about.
Okay.
It was a study case. It was just giving like a hypothetical situation.
Okay.
What you would do. But I
Yeah.
know the question you're talking about. But that one, yeah, it's not on the book. It's just a hypothetical situation.
Okay. I was just a little confused about it. Perfect. Thank you though.
Yeah, if you don't recognize something that was in a book or on the slides, what that probably means is that it's a conceptual question for you to think about and come up with an answer. But look, that's what the discussions section is on Canvas.
Perfect. Okay. Thank you both.
That's what it is for. You just read about it, think about it, write something in the discussion, see what other people think. Right? Now, don't expect the answer from them. So this is something that we need to agree from the very beginning, guys, that we do not provide answers for each other through Canvas, but discussing the general concepts, it's totally appropriate. So that's how we learn. Anything else? All right.
Do you give us like reviews or sample questions to kind of have a gist of how the exam is going to be?
Yeah, I probably will do that. So as I mentioned to you, the best way to study for the test is to look at those objectives, specific objectives, right? So let me start sharing my screen. Right, so chapter 2, water. That's what we're going to talk about today. So on the second slide, here's our specific learning goals, right? And so go over these and study the material which is related to these topics. And that's how I'm going to test you. But you're right. So I will not give you the entire practice exam because it's to come up with so many questions. I'm not so creative. All right. So I need, so I will give you some examples.
Because usually in the past, like with, for example, organic chemistry and all that, I feel like our professors would give us easy questions during lecture, and during the exam they're much harder. So it's, it didn't really correlate to how the exam was going to be. So if you were to give us, for example, a few hard questions for us to solve, it would probably give us a better look at the exam, I guess.
Yeah. The questions. So I have obviously, I've taught this class before, and I have exams from previous years, which I can share with you. But those exams, the new exams will be different, and specifically because I will have to put in quite a bit of time into these new exams because I have to make sure that you cannot Google them, guys, right? So these will not be just a kind of quiz questions where you can just type in the question into Google and find the answer. So these will be more conceptual, more thinking type questions where you actually have to, you'll be presented with the situation and you will have to come up with your own answer. So creating questions like this will take me longer, and these older exams that I have will not be a good representation. So so I will not be sharing the old exams with you because they will give you your own idea of what's going to be on the test. So I will just give some new examples for you. Is there anything else? All right, so here's our learning goals. So water, now you might wonder why we're gonna spend the whole chapter studying a molecule H2O, right, with 3 atoms. So in principle, we can teach the whole course on water. It's a fascinating subject, and the more we learn about, the more scientific advances we make, the more we realize how complex the molecule of water is. It presents so many facets of complexity for us. So we will talk about the type of interactions between molecules. So water provides an ideal environment for molecules to interact. Okay. And then why is it a good medium for life, right? So why did life develop in water, right? So many molecules will be nonpolar, right? So why do they aggregate? A very important part of life is the aggregation of large hydrophobic molecules. And then obviously, if you dissolve something in water, then that will change the properties of water. Okay. For example, here is a question for those of you who like to cook. So if you boil eggs or boil potatoes, if you add some salt to your water, will it make it cook faster or slower?
Faster.
And why is that?
It lowers the boiling temperature.
Oh, well, it's faster because it actually raises the boiling temperature. It raises the boiling temperature, and so the potatoes will boil at higher temperature. And so if they boil at higher temperature, they will cook faster, right? So both okay. So how weak acids and bases behave in water. So we'll talk about acids and bases and buffers, why we need the buffers, and how water participates in biochemical reactions. So we're going to study all those things, and the test at the end will be based on those. Okay, so originally life evolved in water. Well, one explanation is because it protects things from UV light, right? And in general, organisms are basically water, right? So up to 90% of an organism by weight can be just the weight of water. And so there are lots of chemical reactions which can occur in water, right? So water is a very polar solvent. And we know that polar solvents promote chemical reactions because they can stabilize transition states. And also water can get incorporated into proteins, nucleic acids, membranes, and form three-dimensional structures. So what else can we recall from chemistry, general chemistry. We remember the octet rule, remember the oxygen in water is sp3 hybridized, right? 2 hydrogen atoms will be covalently linked to oxygen. And remember, oxygen, which should have 4 electron pairs, will 2 remain non-bonded. So water geometry is a distorted tetrahedron, right? So remember, the molecule of water is bent, right? So if we don't think very hard, somebody tells us, oh, can you draw the structure of water? And if we don't give it much thought, we would draw something like this. And draw something like this. And the teacher will give us that. So water is a bent molecule. And the reason why it's bent is because there are these 2 lone pairs that need space as well. Need space as well. So, and because of that, water will have a dipole moment. Okay, dipole moment, you can serve both as a hydrogen bond donor and acceptor. So the positive charges in the molecule of water will interact with something negative, and oxygen, which is negatively charged part of the molecule of water, will interact with something positive. So remember about hydrogen bonds, right? Hydrogen bonds usually develop between when the hydrogen is attached to an electronegative element, right, such as oxygen, and it will participate in the hydrogen bonding with another oxygen. And remember, these have to be electronegative elements, right? So fluorine will work, nitrogen will work, sulfur will work, but carbon will not work. No carbon here. Okay, no hydrogen bonding with carbon. So water can serve both donor acceptor. Up to 4 hydrogen bonds per molecule. So remember, there are 2 protons, right? And there are 2 lone pairs in the oxygen. So that can give you maximum 4 hydrogen bonds per molecule. That will increase its boiling point, increase its melting point, and also surface tension. So remember, this is the, if you spill some water on that, let's say, table, it will not be a flat, like a spill, not be a flat puddle. There will be edges, right? So the water will adopt a half spherical state. And so that surface tension is due to the interaction between molecules of water. All right, so in general, so this gives you some ideas. So if the covalent HO bond is 420 kilojoules per mole, then the hydrogen bond is about 20, right? So, so that's 1/20th of the covalent bond. Just for us to remember some numbers. And because of that, the hydrogen bond will be much longer, so it's weaker, but it's also longer, right? So these are some distances for us in nanometers. All right, and so hydrogen bond.
Can you
yeah.
Can you explain that? I remember learning that in chemistry, but I forgot what it is. If it's shorter, the bond is stronger, right?
Yeah, the shorter the bond, the stronger it is. Yes. Mm-hmm.
Okay. Thank you.
And it makes sense, right? So how do you break a bond? By making it longer, right? So you stretch the two atoms away from each other until they break, right? And so if they're already far away from each other, then it's even easier to break a bond like that. So right, so we will see this hydrogen bonding everywhere. So unique properties of water, we will see them in proteins, in DNA, right? Polysaccharides, enzymes, hormones, RNA molecules, and some famous saying by Linus Pauling, who is one of the fathers of modern understanding of general chemistry principles, and his book on the nature of the chemical bond. So these are some biological relevance, right? So, so for example, if you dissolve alcohol in water, you will get this kind of hydrogen bond, right? So to think about vodka, right? So vodka is what? Ethanol in water, 40%, right? And when you buy a bottle, do you see two layers? No, you don't see two layers, right? There's a very nice homogeneous single layer, which means that ethanol, so this group R is ethyl, and water mix together without any separation, right? So so ketones, carbonyl of the ketones, so there's a lone pair, will interact with water. Now here's the two amides, right? So this protein here and this protein, so the amide bonds in protein, so there's an NH of one protein will interact with the carbonyl group from another protein. And that's how the proteins will interact with each other and adopt three-dimensional structures, which we will learn a lot about. We have a whole chapter on three-dimensional structure of proteins. And here is the complementary base pairing in DNA, right? So there's adenine and thymine, and you can see very nicely, so there's the two hydrogen bonds nicely aligned with each other to make a base pair in the DNA. You guys have any questions? All right, let's keep moving. All right, so obviously if you freeze water, right, so you will slow down the motion of the molecular motion. Eventually the water will crystallize and it will form hexagonal structures that look like this, right? So, so oxygens of one molecule of water will be hydrogen bonded to hydrogens of a different molecule of water, and so on and so forth. Does anybody know if the density of ice, is it higher or lower than density of water, liquid water?
Lower.
Lower.
And how do we know that? What's the simplest experiment one can do to determine that?
Ice float.
Floats.
Ice floats, that's right. Ice floats. And I guess when the water forms this kind of hexagonal hydrogen bonded structure, it expands slightly, right? It expands slightly, and so there are fewer molecules of water per cubic centimeter or meter of the material, of the structure, right? So if it expands slightly, it becomes a little lighter per certain amount of space, and it floats. All right, so water as a solvent. So we talked about the fact that water is a medium for life, or life developed. And so amino acids and peptides, we will see a lot of charged molecules, charged amino acids. We'll talk about 20 amino acids. Many of these are charged, and water will dissolve. Water loves charged species, right? Water will stabilize the charged species, and it loves dipoles and stuff like that. Small alcohols. What it means is that alcohols that have very short aliphatic chain, very short hydrophobic tail, such as methanol or ethanol. But as you go to higher alcohols, such as butanol, pentanol, octanol, then water is no longer a good solvent, right? Because of the hydrophobicity associated with these tails. And carbohydrates. Carbohydrates have lots of hydroxy groups, so these are alcohols, and they highly water soluble, right? So if you dissolve sugar in water, you know that none of it will precipitate, right? So now water will, in contrast, the water will be poor solvent for nonpolar substances. And so nonpolar gases, aromatic moieties, right? So benzene is not a good molecule for water. Aliphatic chains. So let's look at those. So you can see there's some examples. So here's glucose, right? So glucose, we know that is fully soluble in water. And the reason for that is because of these hydroxy groups, right? So there are 5 hydroxy groups. There's this oxygen. So there's lots of hydrogen bonding available. Now glycine is an amino acid. Now this end is positively charged. This end is negatively charged. And so water loves that. So it will dissolve that. Aspartic acid, the same thing. So there are 3 charges. Lactic acid, there's a charge, there's a hydroxy group. Water will dissolve that. Lactic acid. Glycerol, 3 hydroxy groups, highly soluble. Now, but if you go to lipids, so for example, so typical wax, right? So you can see how long this hydrophobic chain is, right? So this will not dissolve in water, obviously. There is small ester here, which could help a little bit with the water solubility, but is just not sufficient because of the such long hydrophobic chains. Phenylalanine. So now this is amphipathic, which means it has both. It has the water-loving and water-hating functionality. So you can see here there's a benzene ring which will hate water, but then there's charged functionality, right? So phenylalanine is moderately soluble in water because it has both types of groups. Phosphatidylcholine. Phosphatidylcholine is a lipid, which is a component of our membranes, right? So it has a long fatty acid type chain, and it has a charged portion, which is a phosphate and positively charged nitrogen. So in your sapling homework, you will be also asked to determine which molecules are water soluble, which molecules are not water soluble. And the same thing on the test. Okay, So non-covalent interactions. So we've talked about reactions a lot, right? In organic chemistry, mostly. So those reactions involve rearrangement of electrons. But there are physical interactions between molecules, which are important to keep in mind because they also important for life on Earth, and they determine properties of various substances. So here it's important to keep in mind the ionic interactions, right? So we talked about positively and negatively charged. So these are permanently charged species. Also ion and a dipole, right? So one molecule can be an ion and the other can be a dipole. So that's not a possibility. So these will be ionic coulombic interactions. And dipole interactions, so these are just, these are uncharged species, but they're still polar, right? Uncharged but polar. And so the negatively charged ends of those molecules will interact with positively charged and vice versa. Van der Waals interactions. So now here we have attractive dispersion and repulsive, which is steric component. And then there is something called hydrophobic effect. So we'll talk in a moment about hydrophobic effect, but it's a pretty complex phenomenon which is associated with ordering of water around nonpolar substances. Let me see, do I have the, yes, I have hydrophobic effect coming up. Okay. So these are some types of non-covalent interactions. So here we can see hydrogen bond, ionic, right? Now ionic repulsion, the two ammonium groups hate each other. Now hydrophobic interaction, this is where basically these two molecules occupy a pocket, some kind of a pocket, where they're brought together because water repels them. So any two atoms in close proximity. So here, one type of interaction is replaced by another. So here we have ionic interaction, right? Ionic in a table salt, sodium chloride. But then obviously sodium chloride dissolves in water and it will be replaced by ion dipole interaction, right? So the water, which has a strong dipole, will be interacting with the ion chloride in this case. And since there are a whole bunch of those waters, eventually this kind of situation is preferable over the ionic interaction. So the table salt will dissolve because a more favorable interaction is forming. And now you can see here, sodium will also be hydrated, only in this case using the oxygen atoms. So for van der Waals interactions, there are two components. There's attractive force, right? London dispersion forces. And remember, these depend on polarizability. So from organic chemistry 1, and actually even general chemistry, you learned about nonpolar molecules. When they come in close contact with each other, they may induce dipole in each other, right? Even though there's no permanent dipole, but they can interact in such a way that they will induce these instantaneous dipoles, which will result in attractive forces. But if they get too close, then the electrons will repel each other and will create repulsive force, steric repulsion, which depends on the size of the atoms. So attraction at longer distances, repulsion at shorter distances. And so So what you want is you want to optimize this distance for the optimal type of interaction. And these van der Waals interactions are very important, right? So by themselves, they're pretty weak. But in general, they work for any hydrophobic molecule, right? Between any atoms that are near each other. And they're very important for steric complementarity. Now, do I have? No, I don't have it. So let me just draw this. So let's say you have a protein like this, okay? And then you have a drug. You have a drug, and the drug will come in and occupy this pocket in the protein. And let's say this pocket is reserved for some kind of endogenous molecule, for some metabolite that is important for cell viability. And then your drug will inhibit the interaction of this metabolite by taking up this pocket here, right? So in this case, the interaction will be probably in most cases van der Waals interaction. And it's going to be primarily determined by steric complementarity, right? So in other words, this drug has to fit into the pocket. So it's like a key, a lock and key type interaction. Lock and key. Now, so hydrophobic effect is very important to understand. Now you can see that in the past, people would say hydrophobic force or hydrophobic attraction, but not anymore, because our understanding of hydrophobicity has changed. We refer to it as an effect. Refer to it as an effect rather than as a force. So this hydrophobic effect is important for a number of phenomena. For example, protein folding. Okay. So proteins will adopt three-dimensional structures, and a lot of it will be due to this hydrophobic effect. Protein-protein association, right? So the proteins talk to each other. Also hydrophobic effect. Formation of lipid micelles. Can you think of an example of a lipid micelle? Who remembers micelles from general chemistry? Where do we make micelles? Like at home?
What was the question?
Where do we make micelles at home?
Like when you're washing dishes, the soap.
Yep,
Make
the
micelles
soap.
with
That's
the
right.
oil.
That's right. Yep. So, so this soap is a great example of a micelle, right? So I think we'll have a picture of a micelle later. Just wanted to make sure that you're still paying attention. Binding of steroid hormones to their receptors. So also the driving force is its hydrophobic effect. All right, so this does not arise because of some attractive direct force. So it's in fact, it's not an attractive force. Он не поднимешь, а? Okay, so in general, so these are solubilities. You can see that. Just hold on a second, guys. Я просто хотел, чтобы ты выключила телефон. Okay. Okay. So these are solubility of different gases in water. Okay. And you can see here that solubility of gas in water depends on how polar it is, right? So nitrogen is totally non-polar. And you can see now in grams per liter, very little of it will actually dissolve in water. Now, more oxygen will dissolve in water. Now, carbon dioxide is a non-polar molecule, but it has polar bonds, right? So more of it will dissolve in water. But ammonia, a large amount will dissolve in water, and in hydrogen disulfide, so this hydrogen sulfide, a tremendous amount of it will dissolve in water because this has a strong dipole moment. So, but if we move back to our hydrophobic effect, so the hydrophobic effect is actually based on entropy. So if somebody asks you what is hydrophobic effect, the explanation should be based on the entropy. So for example, in this case, when you have just molecules of water by themselves, right? So no interference from any solutes. The molecules of water can adopt different orientation, can adopt different states, adopt different forms. Some of them can form rings. Some of them can be by themselves. Some of them can form hydrogen bonds. So these are like clusters of molecules in bulk phase, right? So this is a high entropy case where basically water does whatever it wants, right? Not limited by anything. Now, if you put a hydrophobic alkyl chain, so this is a fatty acid, okay? So there's a carboxyl group, and then there is a alkyl chain. Then what's going to happen is this alkyl chain, so the molecules of water will have to organize themselves. They will have to create an order around this hydrophobic alkyl chain, right? And by doing so, it reduces the entropy. Remember, entropy, if you reduce entropy, the energy of the system increases, right? Remember, delta G equals delta H minus T delta S. So you want the entropy to be larger for the energy to go down. For the delta G to be negative, you want the entropy to be larger, right? Let me write this down. Delta G equals delta H minus T delta S, okay? So you want this term here, S, to be large. If S is large, then delta G is more negative, which will bring down the energy of the system. So when the system is highly organized, the entropy goes down. The entropy goes down, and then delta G is not so negative anymore. Okay, so it's a low entropy case. And now you put this fatty acid in the water, right? So water will organize itself around this fatty acid. It will organize itself around this fatty acid. It will organize itself around this fatty acid, right? So this is actually a little bit of good news. There's some carboxylic acid here interacting with water, some positive ionic interaction. But in general, what will happen is that these molecules will undergo aggregation, right? Go back. They all by themselves. Look how many waters are necessary to surround these fatty acids. Now these fatty acids come together, and now you need many fewer molecules of water to organize themselves around these fatty acids. All these other molecules of water are released, and they can do whatever they want, right? So the entropy of the system will increase. So fewer molecules of water are ordered, and entropy is increased. So this is what the hydrophobic effect is about, right? So this is what causes biological membranes to form. All right, so we talk about biological membranes in a cell. We talk about a lot, you read about it in chapter 1. And what causes this biological membrane formation? It's the entropy, right? It's the entropy which causes these fatty acids to come together and form a membrane. Ты не принесешь мне этот высморкаться? Высморкаться. Okay, any questions about entropy, about hydrophobic effect?
Can you repeat that last part about when delta G is more negative? You said that it brings the energy of the system up, or did I not understand it correctly?
So you want the delta G to be negative, right? So you want, so any process that will reduce the energy, which will make delta G more negative, is going to be favorable. It's going to be spontaneous.
Okay, thank you.
And so that will occur if your delta S is positive because of this negative sign here. Okay, and here's, so these are membranes, but if the concentration of these amphipathic molecules is sufficiently high, it can aggregate all the way into micelles. Micelles, so you can see here, the polar head groups are going to be on the outside, and the non-polar alkyl groups are going to be looking inside of the micelle. And so this create, for example, in biological systems, this create what's known as vesicles. Vesicles, for example, some of the vesicles you read about, vacuoles in plant cells. Various cellular organelles are based on the formation of vesicles. Okay. And obviously this hydrophobic effect is important. I drew the picture for you how a drug would bind to a receptor, to a protein in the body. And so the driving force for that will be hydrophobic effect, right? So, so the drug, because the drug is exposed to aqueous medium, will want to escape the aqueous medium, and instead it wants to actually come in close contact with the hydrophobic large surface of the protein and kind of hide away from the molecules of water and increase the entropy by displacing water from the protein. Yeah, that's shown here, right? So here's the enzyme. Here's the molecules of water which are organized. This is the substrate or the drug. The substrate comes in, interacts with the enzyme, and you can see this the stickiness. You can see the two hydrophobic surfaces are interacting with each other, and the driving force is the displacement of all these molecules of water which can come off, and the result of that will be the increase of the entropy. Okay, so let's see where we stand. Sure, we'll keep the annotations. We are on page 30, 53. We can keep going, I think, at this pace. We still have time. So, any questions so far, guys? Any questions about anything we've talked about?
So for the structures that you gave us earlier in the PowerPoint, are you going to test us on that, or was that just solely to explain what you were talking about?
Like what structures?
Let me pull it up. I don't even remember. It was like earlier in the PowerPoint.
This?
No, like the organic structures.
Organic.
Yeah, these like glucose.
No, I want you to, well, I may not be able to draw, I may not give you these specific structures, but I would like you to recognize which ones are polar, which ones are non-polar.
Okay.
And which ones will make things water soluble and which ones will make things hydrophobic.
Okay.
Yeah, so if you see something with a long alkyl chain, I want you to be able to see that and tell me, yes, this molecule will not dissolve in water because of the long alkyl chain.
Okay.
So just like water changes the properties of dissolved substances, the substances can change the properties of water. It's important to realize. Now there is this colligative properties. So we talked about boiling point, right? So put sodium chloride, boiling point changes. Talked about melting point. What happens, by the way, if you put some sodium chloride in water? Will the melting point decrease or increase? So why do they put, okay, so maybe Texas is not a great example, but if you live in Detroit or Boston, they would put salt on the road in the winter. Why would they do that?
To melt the ice.
That's right, it will melt the ice. And why would it melt the ice? So it will change, obviously, by melting, it will change the melting point of water. So will it make melting point higher or lower?
Lower.
Lower, correct. So so if you put salt in water, the salt, so the water will freeze at a lower temperature, right? And can be as low as -20. In fact, in a chemical lab, we often use this trick as well. If you need to run a reaction and 0 degrees, let's say you run a reaction at 0, you use an ice bath. But if you want to run your reaction even at a lower temperature, you would put some salt in your ice bath together with your water, and you can lower the temperature to, let's say, -18, for example. Okay, and there is non-colligative, viscosity, surface tension, taste and odor. Makes sense. Okay, in the cytoplasm of cells, highly concentrated solutions because of the osmotic pressure. Right, so remember osmotic pressure, right? It's a movement of water in a direction towards the highest salt concentration. And the pressure to resist that is called the osmotic pressure. So it's shown here, right? So this is pure water. Now here there's a filter or membrane, right? And we're gonna put some salt in this test tube. And because of that, the water will come in through this membrane. The salt cannot escape, but the water can come in and it'll raise the amount of water. It'll increase the amount of water in the test tube because the salt will want to be diluted. And you will need to apply pressure to put it back down where it came from. And that is actually the osmotic pressure. And what does that mean in terms of cells? So here is the, let's say if your cell in an isotonic environment, right? What that means is that the amount of salt outside of the cell is the same as the amount of salt inside the cell. And so there's no net movement of water, no osmotic pressure. But if you put a cell in a hypertonic solution, okay, so which means it's much more concentrated, so then water will come out. Water will come out to dissolve all the salt around it, and the cell will shrink. Like, can you give me an example of this process in daily life? Where do you see something like this happens?
Dehydration.
So when we are dehydrated, we shrink. Maybe, I don't know. I'm just not sure.
Sickle cell anemia.
Sickle cell anemia. But that's the irregular shape of the red blood cells.
Oh, yeah, you're right.
That happens if you put a freshwater fish in a saltwater environment.
Okay. So the fish will get smaller.
Mm-hmm. That's why you can't switch. That's why they can't live in saltwater.
Yeah, that's true. How about okay, I'm going to give the answer away. How about a pickle? That's what it is, right? So you take a piece of cucumber, you put it in a highly hypertonic environment with lots of salt and vinegar, and it will shrink. Now, the opposite is true too. So if you put a cell in a now it's hypotonic environment, okay, where actually there is less salt on the outside, then the water will come in. the water will come in to dilute all the salt inside, in which case the cell may burst, right? And so we talked about bacteria a little bit last time. Well, we didn't talk about bacteria last time, but you read in the textbook that bacteria have cell walls, right? Gram-positive bacteria. And the cell wall is designed to protect the bacterial cell from osmotic changes in osmotic pressure. And so the bacteria is designed to survive in different environments, in the hypertonic environment and the hypotonic environment. And the cell wall, it's part of the function of the cell wall is to keep the cell stable under different environments. So just to remind you, there's some math in your sapling. So let's quickly go over that. So remember that water dissociates, right? A little bit dissociates to form protons and hydroxide ions, but it dissociates very little, right? So the equilibrium is strongly shifted to the left. And there's equilibrium constant for that, right? And remember, when you draw H plus in water, you should draw it as hydronium ion, right? So H plus does not live by itself in water. It lives in the form of a hydronium ion. So basically it'll bind to another molecule of water and through the process known as solvation or hydration. And this process is known as proton hopping, right? So for example, if you have hydronium ion, then another molecule of water can come pick it up. At the same time, another can pick it up, and it can actually build into these interesting structures where this proton is not here, not here, not there, not there. It's everywhere, right? So all these molecules of water are participating in sharing this proton. And so the proton is everywhere. Can't find it. So it's known as proton hopping. Okay, so one important thing here is that the equilibrium constant for this can be simplified a little bit. Now, this has been determined experimentally, and the water can be determined from water density. So in other words, there is 55.5 moles in 1 liter of pure water. Okay, so if you plug this in, you can simplify this equation to this. And since the concentration of protons and hydroxide ions is the same, right? There's no other way. Molecule of water, if it dissociates, it will create the same number of protons as there are hydroxides. So then you can divide this in half, and then you can get the concentration of protons in pure water, which is 10 to the negative 7th. 10 to the negative 7th. Then you take this equation, take the negative log of both sides, and then you come up with the simplified equation where the pH and pOH equals plus 14. So it's a much easier version to work with, right? Just remember that pH is negative log of the concentration of protons. So the neutral solution, these two are the same and equals 7. So very easy to work. So pH varies, right? And because this is negative log, so you can see this is the actual concentration of protons, and this is the pH, right? So if you take the log, negative log of this, you get 0. So pH, some common liquids, you can read about it. So let's look at some specific example of dissociation of a weak electrolyte. So here we have acetic acid. So it's a weak acid, okay? And so the equilibrium constant for its dissociation is the multiplication of the two products, which is the proton and the conjugate base. And in the denominator, we have the concentration of the starting concentration of the acetic acid. And this is the equilibrium constant given to us. And let's look at some examples of some problems one can be faced with. So let's say you have a solution of acetic acid, 0.1 moles. And then you add that to water. So you add 0.1 moles of acetic acid to water so that the final concentration is, so the final volume is 1 liter. So it's pretty straightforward. You just pick an X, which is the concentration of the proton, which is what we're looking for, right? The concentration of the conjugate base will be the same because each proton will be produced together with 1 molecule of conjugate base. And if we started with 0.1 molar, then the equilibrium concentration will be 0.1 minus X. So here we write it down, this equation. So this is a quadratic equation, we solve that, and we get the concentration of protons, and take the negative log that, and we get concentration, we get pH of 2.8. So keep in mind for pHs, remember the pH of the neutral pH is 7, right? If pH is less than 7, the solution is acidic. Concentration, if the pH is higher than 7, the solution is basic. And if this is something that still kind of gives you some problems, general chemistry could be a great resource, right? So we solve these equations in general chemistry and even in organic chemistry as well. Now this, if you don't want to deal with the quadratic equation, if we go back, keep in mind that a very small amount of acetic acid will dissociate. So the X is much smaller than 0.1. What that means, you can actually take it out of here, right? Just totally neglect it because such a small number. And so in this case, in the denominator, you will have 0.1. And so now this is not the quadratic equation anymore, very simple equation. And you can see the pH is exactly the same as what we calculated before. Yeah. So it's a very nice approximation that you can take advantage of. One last thing I want to talk about in this lecture is buffers, right? So biochemistry is all about buffers. So we want our proteins to keep their three-dimensional structure. And so if you change the pH of the solution, you can unfold the protein can change its shape, you can kill a protein, right? You can give it all different function to a protein by changing the pH. And so to prevent that from happening, you use buffers. So you work with proteins and buffers. You probably heard things like phosphate buffer, right? A carbonate buffer. So what are those? So here is the buffer. So basically this is the solution of a one-to-one mixture of acetic acid and some kind of acetate. I think it's sodium acetate in most cases. Sodium acetate. And then basically what you're going to do, you're just going to start with the acetic acid on the left, and you're going to start adding the hydroxide ion. And so hydroxide ion will convert acetic acid into acetate. And as you do that, you increase the number of equivalents of OH minus that you add, okay? And in the process, the pH will rise, right? So you start with the low pH, but since you're gonna be adding base, the pH will rise towards basic pH. And so eventually, there's this equivalence point. After you've added half of the amount of OH, of hydroxide ion, you will have reached the pH of 4.76, which is known as a pKa value for this acid. And you can see here, so this region between pH 3.76 and 5.76, it's known as a buffering region, okay? So you can see actually it's pretty resilient, right? So you can actually play with the OH as much as you want. You can add lots of OH minus here, but the pH will not change tremendously. And so people say that the loading capacity of the buffer is very high. So you can add lots of OH minus, but the pH will only change by 2 units. Okay. So please read about buffers in the textbook and some problems in sapling on buffers. And now there are other kind of buffers. So look at that as well. Very useful equation. You may remember the name of it, Henderson-Hasselbalch equation. So which allows you to determine pH values for any amount of acid present. Okay. And so I think there are a couple of problems in sapling on the use of this equation. So in this case, you will make a buffer. You will take 0.1 mole acetic acid. pKa is known. You take 0.05 molar sodium acetate, mix them together, plug this in, and it will give you the buffer. It will give you the pH of this buffer, 4.45. And obviously, we did not invent buffers, right? So the buffers were biological systems utilize buffers long before we came. And you can see here. So the intracellular pH is vital to cells. And for example, bicarbonate buffer is one of the main ones that the bodies utilize. There's phosphate buffer, histidine buffer, and finally, there's a couple of slides showing that water can not only be a solvent, it can be part of a three-dimensional structure of different proteins, right? So you can see here, so this protein shown here has water incorporated into it and allows to make contacts between different side chains, amino acid side chains, using water molecules as bridges, right? So for example, you want to make a hydrogen bond. Let me do this. Let's say you want to make a hydrogen bond between this proton and this carbonyl, but they're too far away from each other, right? And so water can be used as a bridge. You can insert a molecule of water, and you can see now there's this oxygen interacting with the proton, this proton interacting with this oxygen, and there is a very nice hydrogen bond. So these two hydrogen bonds that you just created allowed for the formation of this three-dimensional structure in the protein. Okay, guys, you can wake up now. You can wake up now. Now, as far as chat, let's see what we have. What about grapes? Grapes.
That was in response to the question about the osmotic pressure.
So,
I guess they were thinking raisins.
oh,
Maybe.
raisins. Yeah. But that's more. Yeah, so basically grapes will shrink, right, into raisins. That's what you're trying to say, right?
Yeah, that's what I was going for.
Yeah. Okay. All right, so the homework is due. I'll make an announcement on that. I think the homework is due on Tuesday, but I'll double check on that and make it.
I'm sorry. Is the chapter 2 homework open now?
Not yet. I will open it.
Okay.
Good questions. I'm going to open after this class.
Perfect. Thank you.
But chapter 1 homework, that is due Friday, if I'm not mistaken. Let me just check. Sapling. So chapter 1 homework, August 28, 9 o'clock, that's this Friday. And chapter 2, which I'll make available after this class, is going to be available until September 2nd. And September 2nd is...
That'll be next Wednesday.
That's Wednesday, yeah. Okay, guys. Sounds good. And I would like to see more activity in a discussion on Canvas. All right. Stay safe.
Thank you, sir.